I'm going to introduce Tanner Vandever. He is a new master's student in the Department of Biology. He started out as an undergrad taking my genetics course as well, and really was in the psychology department. I was like, dude, you got to get out of here. This is genetics or biology. He's like, no, really, I have an interest. So he transitioned into the lab just one semester and then kind of stuck around. He has interest in neurodegeneration, and so his project today, he's going to tell you about it, but it definitely shows that toxins in the environment can lead to potential susceptibility for neurodegeneration. Tanner?
Thank you all for your time. My poster is on the investigation of BMAA modulation of amyloid beta-mediated neurodegeneration and transgenic C. elegans. I myself am Tanner Vandever. My co-authors are Elise Patrick and Alexis Petty, along with some wonderful help of Dr. Ashley Turner. So to start us off, BMAA, beta-N-methylamino L-alanine, is a non-protein amino acid and neurotoxin originally isolated from seeds of the cycad plant found on an island, I believe around the Guam area, in a village of people called the Caenorhabditis people. This was first discovered by the US military around the 1950s and 60s, and once they started identifying that these people were having an issue, studies picked up a little bit and then eventually dropped off over time and have picked back up since then. The main introduction of BMAA into the Caenorhabditis people had been through their diet, through a process called biomagnification. So as the wildlife, specifically bats in this case, when they consume cycad seeds or the fruit, etc., it builds up in the bat, and then the people will eat the bat, causing it to biomagnify. So the symptoms of BMAA accumulation are very peculiar. They kind of come in a form of a combination that they titled ALS-PD complex, which is amyotrophic lateral sclerosis / Parkinsonism dementia complex. So it's a huge group of a bunch of different mental illnesses all packed into one. And unfortunately, the life expectancy of the Caenorhabditis people, you can say, is probably about 50 to 60 years old. So my group and I, we plan to do a comparison of BMAA neurotoxicity to human Alzheimer's disease as they present in a similar way. In human Alzheimer's disease, we see an accumulation of a plaque on neurofibrillary tangles of this amyloid beta, and it gets worse as you get older because these amyloid beta molecules will build up on the tangles, and the body eventually loses the ability to get rid of them altogether. So our hypothesis is if transgenic C. elegans nematodes expressing human amyloid beta are exposed to BMAA, then the treated transgenic animals display more severe neuromuscular defects, shortened lifespan, and age-dependent behavioral dysfunction compared to untreated wild-type N2 animals. And N2 is our wild-type nematode strain here. So for our materials and methods, starting off, we had Microsoft OneNote, Teams, OneDrive, just for communication, data entry, stuff like that. We have our C. elegans AD strains. These are human amyloid beta expressing strains, which are GRU101 and GRU102. We have our BMAA. We have our homes for our nematodes, which are petri dishes and growth media reagents. And we will also be using a dissecting microscope with a mounted camera for viewing, manipulating, and recording nematodes. found this really nice 3D model graphic right here. I know it might be hard to see on the poster, but this is a visualization of the neurons throughout a nematode's body. And that's what makes nematodes such a great model for neurodegeneration and also a lot of other human diseases. They have an oddly complex neuron system for being such a small animal, and it's absolutely amazing to me that we can manipulate neurons and compare them microscopically into a translation of the human body. So for our experimental design, we're going to do an initial Alzheimer's disease nematode phenotyping, and we have split that out into 3 different assays. We have a thrashing assay, and in nematodes, they have a very unique behavior in which when you drop them in a liquid, they begin to thrash around, just sporadically. And using this as an assay, we can compare, let's say, wild type N2, we can see how much they thrash around in these liquids, and compare them to, let's say, Alzheimer's disease or the strain that's been treated with the BMAA. We can compare how the neuromuscular system is beginning to degenerate as they begin to thrash less. We have a lifespan assay as well, so just to see how short of a lifespan these nematodes might experience, and of course comparing those to the N2, and then a behavioral assay, which we had tossed around the idea of, but you can train nematodes to go towards negative stimuli. So the thought in this case would be, if the nematode has a large amount of neurodegeneration, can it understand whether or not the negative stimuli is negative? Will it begin to move towards negative stimuli instead of beneficial stimuli? Will it be able to tell the difference? So here's a breakdown of the different groups that we'll be moving forward with in this design. We have our wild type N2 for our control. We have a transgenic amyloid beta strain. We have a transgenic control strain. And then down here is the listed vehicles or the bacterial clones that we'll be using as vectors to move these genes into the nematodes, which is a wild type N2 vehicle, a transgenic amyloid beta strain vehicle, and a transgenic control vehicle. And lastly, for this graphic, we are still figuring out which assay we would like to move forward with. We do have a couple other experiments that we've done where we've tried a lifespan assay. I don't remember how far it got off the ground, but we do have an interest towards the thrashing assay as well, so we'll be giving both of those a try. As a part of my discussion here, nematodes serve as a great model to examine the impact of BMAA has on amyloid beta-mediated neurotoxicity and neurodegeneration. I had mentioned this earlier, but the fact that nematodes have such a complex neuronal system makes them great models to compare human neurodegenerative diseases. BMAA accumulation has been found in individuals living near bodies of fresh water. And this was from a paper of comparison between dead Canadian brains and Comoran brains. And when they did the brain analysis on the tissue, they couldn't tell the difference. They had no idea which was which. So here we get a glimpse at the potential scale of neurotoxins in our environment, not just BMAA and these blue-green algae, these cyanobacteria, but what other neurotoxins or what other things are being brought in from our environment that we don't know about. This will give us great insight into those pieces. And lastly, I would like to do some acknowledgements towards the Department of Biology and as well as Sir Paul Cox. He was the one that provided us with the initial sample of BMAA. And just a quick view of the future directions here. we'll do our initial experiments covering the 3 first assays. And then once we get a baseline from that, we'll move forward with BMAA introduction. Thank you all for your time. Please.
How do you train a nematode?
I suppose you would do that the same way that you would train any animal. So if it does a behavior, you give it positive reinforcement, and it will make an association to, I like to go there because I know something good happens when I go to that spot, versus something that's bad, or just as an instinct. So if something, I guess you could say, smells good, you'll want to move towards that, and you'll want to move away from something that smells bad.
Other questions?
So you said that BMAA is extremely toxic. How do you plan on using that in the lab and not infecting yourself or people who work around you?
We will be doing all of our experiments with PPE, under a hood, and this is also in a powder form. So it's not like I'm going to be able to get it on my skin and absorbs, or if I'm wearing PPE, I'm not going to breathe it in. It'll all be done in a safe environment under a hood and away from potential neurotoxicity.
That's great.
What else?
All right, let's thank our speaker again.